Bro, Mars called, and it's not dead. Number 10. Hydrothermal deposits. 3.5 billion years ago, parts of Mars looked a lot like Western Australia does today. Hot water pushing through cracks in the ground, pooling in shallow basins, leaving behind layers of silica and carbonate that stack up like pages in a book. In the Pilbara region of Australia, those same mineral layers hold the oldest confirmed fossils on the planet. Tiny microbial mats pressed between sheets of geyserite, preserved so well that researchers like Martin van Kranendonk at the University of New South Wales can read the texture of individual cell colonies under a microscope. Mars has its own version. Orbital instruments on the Mars Reconnaissance Orbiter have mapped carbonate and silica deposits in several ancient basins, including areas around Jezero Crater and the Nili Fossae region. The mineral signatures match what forms when hot water loaded with dissolved elements hits colder surface water and dumps its chemical cargo. What matters is what those deposits can trap. Hot spring environments concentrate organic molecules the way a net concentrates fish. Dissolved nutrients flow through the rock, and anything living in the water gets entombed in mineral layers as they form. The Pilbara fossils survived 3.5 billion years because minerals locked them in place before decay could finish them off. If the same process ran on Mars, any organisms living near those vents would have been sealed into mineral pages that the planet then buried under dust and basalt. The chemistry is a match. The minerals are a match. The only missing piece is the biology itself. And that is a question no orbiter can settle from orbit. Number 9. Organic carbon. In 2022, Perseverance was drilling into mudstone at the western edge of Jezero Crater's ancient delta when the Sherlock instrument flagged something the team had been hoping for but did not expect to find so cleanly. A strong signal for organic carbon, not in one spot but scattered across multiple drill targets in the same sedimentary layer. Sherlock works by firing an ultraviolet laser at rock and reading the way molecules scatter the light. Its principal investigator, Luther Beagle, and the team at JPL had designed it specifically to hunt organic signatures. When the readings came back positive from several spots in the same fine-grained mudstone, the room got quiet. Carbon is the backbone of every living molecule we know of. Finding it locked inside ancient lake mud is significant because of how the mud behaves. Clay and silt settle slowly in calm water. As they pile up, they bury whatever is mixed into them, sealing it from ultraviolet radiation and chemical breakdown. That is the same mechanism that preserves organic traces in terrestrial lake sediments for billions of years. The carbon Perseverance found is not sitting on the surface where a stray meteorite or a photochemical reaction could explain it. It is embedded in the fabric of the rock, mixed into the mineral grains in a way that suggests it was there when the sediment formed. There are non-biological ways to get carbon into mud. Meteorites deliver organic material constantly. Volcanic outgassing can produce carbon compounds that wash into lakes. But the pattern here, carbon distributed through multiple layers of quiet lake sediment rather than concentrated in a single impact spot, is harder to explain that way. Whatever put the carbon there did it repeatedly, over time, across a wide area. That is not what a one-time delivery looks like, it is what a biologically active lake looks like, and that is what makes the mudstone at Jezero one of the most closely watched pieces of rock in the solar system. Number 8. Redox chemistry. Abigail Allwood spent years studying ancient stromatolites in the Pilbara before she became the principal investigator for Pixel, the X-ray instrument bolted to the end of Perseverance's robotic arm. Pixel maps the chemical makeup of rock surfaces at the scale of a grain of sand. What it found inside the Jezero mudstone looked familiar to her. Iron and sulfur were not evenly mixed. They formed gradients, zones where iron shifted from one chemical state to another and where sulfur concentrated in narrow bands. Those gradients are not random. They represent stored chemical energy, the same kind of energy that microbes harvest in deep ocean mud and underground aquifers. Think of it like a battery embedded in the rock. One side holds reduced iron, the other holds oxidized compounds. The difference between them is potential energy. Bacteria in terrestrial sediments make their living by bridging that gap, moving electrons from one side to the other and capturing the energy released. Pixel also detected phosphorus concentrated in the same layers. Phosphorus is not optional for life. It is the backbone of DNA, the currency of cellular energy, and a building block of cell membranes. Finding it alongside iron and sulfur gradients in a fine-grained lakebed deposit is the chemical equivalent of finding a stocked kitchen with every ingredient a cook would need. The tricky part is proving someone was cooking. Water moving through porous rock over billions of years can set up gradients without any help from biology. Minerals react slowly with dissolved compounds and sort themselves into layers. The question is whether the arrangement, pixel measured, matches what geology does on its own, or whether it lines up better with what microbial metabolism produces. Allwood's team is comparing the Jezero patterns to both biological and non-biological examples from terrestrial sediments. The answer is not obvious yet, but the rocks have all the ingredients and all the energy a microbial community would need. Whether anyone was home to use them is the open question. Number 7. Manganese oxide. Curiosity was scanning rocks in Gale Crater when its laser hit something the mission team had not predicted. Concentrations of manganese oxide far higher than any model had called for. Manganese oxide means one thing almost without exception. Oxygen and liquid water work together to pull manganese out of solution and lock it into rock. That process plays out in lake beds and river deltas where water carries dissolved manganese until conditions change. The water turns more oxidizing, the manganese drops out, and dark layers of oxide build up over time. Microbes often drive that reaction. Manganese oxidizing bacteria use the metal as an energy source, stripping electrons from dissolved manganese and leaving solid oxide behind. It is one of the oldest metabolic strategies in the microbial playbook. Finding that chemistry on Mars raises an immediate question. Where did the oxygen come from? The Martian atmosphere today is 95% carbon dioxide with barely a trace of free oxygen. 3.5 billion years ago, the picture was supposedly the same, or even more reducing. Yet here are deposits that require strongly oxidizing conditions to form. One possibility is that ancient Mars had bursts of atmospheric oxygen nobody realized. If photosynthetic microbes ever lived in Martian lakes, they could have produced oxygen as a waste product, the same way cyanobacteria did on early Earth. Those pulses would have been local and temporary, building up in lake water before dissipating. But they would have been enough to oxidize manganese and leave exactly what Curiosity found. Non-biological oxidants could also do the work. Some researchers have proposed that halogens like bromate dissolved in acidic water could oxidize manganese without any oxygen at all. Ultraviolet radiation hitting the surface might also generate reactive oxygen species, but those models struggle to explain the sheer concentration in the rock. The deposits are not faint traces. They are rich seams that match the kind of buildup you see in biologically active lakes back home. So far, no purely geological model fully accounts for what Curiosity measured. A chemical fingerprint that back home would be strong evidence of microbial activity sitting on a world where microbes have never been confirmed. Number 6. Cauliflower silica. In 2007, Spirit was exploring the Columbia Hills region inside Gusev crater when it photographed something that looked out of place. Knobby, cauliflower-shaped nodules of opaline silica rising from the soil near a volcanic feature called home plate. Opaline silica forms when hot water, rich in dissolved silica, cools and precipitates. You find it around hot springs, geysers, and hydrothermal vents. The shapes Spirit captured were not smooth or crystalline. They had bumpy, digitate, finger-like protrusions that looked organic in the literal sense. They looked like something had grown them. Stephen Ruff and Jack Farmer at Arizona State University spent years comparing the Martian structures to deposits at hot springs around the world. They focused on El Tatio in the Atacama Desert of Chile, one of the highest and driest geyser fields on the planet and the closest analog to Martian surface conditions. The silica at El Tatio is nearly identical in shape, size, and texture to what Spirit photographed. There, those structures are built by microbial communities. Colonies of heat-loving bacteria and archaea form mats on the silica surfaces. As hot mineral-laden water flows over the mats, silica precipitates around the microbes, entombing them layer by layer. The result is those distinctive cauliflower shapes, microstromatolites that preserve biological textures in stone. Ruff and Farmer also checked deposits at Yellowstone and the Taupo volcanic zone in New Zealand. In every terrestrial case, the cauliflower morphology was tied to microbial activity. No purely abiotic process in any of those environments produced the same shapes. That does not prove the Martian structures are biological. It proves that every known example of that exact shape involves life. Home plate was once a hydrothermal system driven by volcanic heat. Hot water moved through the ground, carrying dissolved minerals and creating a warm, wet, energy-rich setting where life thrives on every continent. If microbial life ever existed on Mars, a volcanic hot spring is one of the most likely places it would have lived and one of the best places to look for its remains. Spirit lacked the instruments to read biosignatures at the molecular level. It could identify the mineral, but not the microscopic textures that would close the case. A structure that looks biological sits where biology would thrive and matches biological formations in every measurable way except the one that matters most. Number 5. Leopard spots. In 2024, Perseverance drilled into a rust-colored boulder nicknamed Chayava Falls near the inner rim of Jezero Crater. When the Sherlock and Pixel instruments scanned the core, the readings puzzled the team at JPL. The rock was pitted with dark rings and tiny nodules that the team began calling leopard spots. Inside those spots, two minerals appeared together that had never been found side by side on Mars: vivianite, an iron phosphate, and greigite, an iron sulfide. Vivianite typically grows in waterlogged sediments where organic matter is actively decomposing. Bacteria break down dead tissue, releasing phosphorus that combines with dissolved iron in the surrounding mud. The result is vivianite crystals embedded in organic, rich clay. Greigite tells a related story. It forms in low-oxygen muds where sulfate-reducing bacteria pull sulfur from the water and lock it into iron compounds. The two minerals appearing together is a signature that microbial communities in terrestrial wetlands leave behind routinely. Sunanda Sharma and the Sherlock team also detected organic carbon concentrated inside the spotted zones, not scattered randomly through the rock, but focused in the same spots where the vivianite and greigite sit. That association matters because it mirrors what happens in biologically active muds. Microbes cluster, they metabolize, they change the chemistry around them, and the mineral evidence of that activity stays locked in place. The team tested alternatives. Hot fluids moving through the rock, simple water chemistry concentrating organics by coincidence, radiation or weathering creating the spotted pattern on its own. Every alternative fell short on at least one count. Either it could not produce both minerals together, or it could not explain the organic carbon in the same locations. That does not mean the answer is life. It means the answer is not obviously geology either. Perseverance has sealed a sample of Chayava Falls in one of its cache tubes for a future retrieval mission. The leopard spots are a pattern that biology makes easily and chemistry struggles to fake. Whether Mars managed the trick without biology is the question sitting inside that tube. Number 4. Complex organic molecules. In 2018, NASA announced that Curiosity had found something in 3.5 billion-year-old mudstone at the base of Mount Sharp that changed the conversation about Mars. Complex organic molecules including sulfur, bearing compounds called thiophenies and aromatic hydrocarbons like benzene locked inside ancient lake sediments. These are not simple carbon scraps. They are structured molecules associated with biological decay, the breakdown of cell membranes, the residue of chlorophyll, the chemical aftermath of ancient life. Finding them preserved for billions of years in Martian rock was a turning point. The discovery deepened in 2026. Curiosity used a wet chemistry experiment for the first time on Mars, treating a drilled sample from the Marianning site with a reagent called tetramethylammonium hydroxide. 21 distinct organic molecules appeared, 7 of them never before detected on the planet. Among the new finds were nitrogen-containing heterocycles, ring-shaped molecules that serve as precursors to the nucleobases in RNA and DNA. The same class of molecules that carry genetic information in every living thing showed up in Martian dirt. Alongside them was benzothiophene, a sulfur-bearing molecule known from carbonaceous meteorites and considered a marker of prebiotic chemistry. What makes these findings hard to dismiss is where they were sitting, not on the surface where meteorite dust or ultraviolet reactions could account for them. Buried inside clay-rich sediments that settled in calm lake water and were sealed by rapid burial. That is exactly how ancient organics survive on any rocky world, locked in fine-grained mud where oxygen cannot reach them and radiation cannot tear them apart. The sulfur in thiophenies acts like a chemical shield, protecting carbon chains from the radiation that hammers the Martian surface. That explains how delicate molecules survived 3.5 billion years, but it does not explain where they came from. Volcanic processes, meteorite delivery, and certain water rock reactions can produce some of these compounds without biology, but the diversity and the specific types, particularly the nitrogen heterocycles, are harder to explain through purely geological chemistry. In terrestrial settings, those molecules almost always trace back to living systems. Number 3. Methane spikes. Something on Mars is releasing methane, and nobody can say what. Curiosity's sample analysis at Mars Instrument has been sniffing the air inside Gale Crater since 2012. What it found does not fit any simple model. Background methane rises and falls with the seasons. It climbs at night and drops during the day. Then, without warning, the readings spike to levels 40 times higher than the baseline. One moment the air is nearly clean. The next, it carries a pulse of gas that in a terrestrial atmosphere would almost certainly point to biology. Roughly 95% of methane in our atmosphere comes from living things. Cattle, wetland microbes, even termites. The rest comes from geology, mostly water reacting with hot rock deep underground. Mars has no cattle and no wetlands. So where is it coming from? That is the puzzle. The European Space Agency sent the Trace Gas Orbiter specifically to map methane from orbit. It found almost nothing. The gas shows up at ground level but vanishes before it reaches the upper atmosphere. Something is destroying or trapping it on a timescale too fast for known chemistry to explain. One team at NASA Goddard suggested the methane sits trapped beneath crusts of solidified salt in the soil. Temperature shifts or even the weight of the rover rolling over the ground could crack those seals and release concentrated puffs. Another group at Johns Hopkins showed that changes in air pressure driven by solar heating could make the ground breathe, pulling gas up through fractures at night and pushing it back down during the day. Both ideas explain the timing. Neither explains the source. The methane has to come from somewhere underground, either from ancient rock chemistry or from something alive. Subsurface microbes called methanogens thrive in similar conditions back home, deep in rock, far from sunlight, eating hydrogen and carbon dioxide and exhaling methane. If such organisms ever lived beneath Mars, they could still be producing gas today, or the methane could be a fossil leak from a biosphere that died billions of years ago. Either answer would reshape everything we know about life beyond this planet. No instrument on the surface can tell biological methane from geological methane. That requires isotope measurements precise enough to read the carbon-13 to carbon-12 ratio, a level of precision no rover carries. Until someone gets that reading, the spikes remain one of the most unexplained signals coming from a planet we keep calling dead. Number 2. ALH 84001. In 1984, a team hunting for meteorites on the blue ice fields of Allen Hills in Antarctica picked up a potato-sized rock that would change the trajectory of space exploration. 12 years later, NASA held a press conference that made global headlines. The rock, cataloged as ALH 84001, was a piece of Mars blasted off the surface by an ancient impact roughly 17 million years ago. It drifted through space and landed in Antarctica about 13,000 years ago. Inside it, a team led by David McKay found 4 features that together looked like a calling card for Martian microbes. First, tiny carbonate globules arranged in layers that suggested liquid water had moved through the rock around 3.9 billion years ago when Mars was warmer and wetter. Second, inside those globules, the team found grains of magnetite and iron sulfide sitting side by side in a pattern produced almost exclusively by magnetotactic bacteria. These organisms build chains of magnetite crystals inside their cells to orient themselves using magnetic fields. Third, complex organic molecules called polycyclic aromatic hydrocarbons appeared concentrated around the carbonate globules. Those molecules can be decay products of dead organisms. Fourth, electron microscope images revealed elongated structures roughly 100 nanometers long that looked startlingly like tiny worm-shaped fossils. The announcement triggered one of the fiercest scientific debates of the 20th century. Critics argued that the magnetite could form without life through high-temperature reactions. They said the organic molecules might be contamination from Antarctic ice that seeped into the rock over thousands of years. They pointed out that the worm shapes were far smaller than any known cell. Supporters fired back. They showed that the magnetite chains had a purity in crystal geometry that matched living bacteria and not laboratory-grown crystals. They demonstrated that the organics increased toward the center of the rock, the opposite of what contamination would produce. And they noted that the size argument depended on assumptions about minimum cell size that later discoveries of ultra-small bacteria have challenged. Decades later, the debate still has no winner. Studies in the 2020s re-examined the magnetite with advanced instruments and found features that remain hard to explain without biology. Other work showed that some of the structures can form through purely geological processes under specific conditions. What makes this rock so important is not that it proves life. It is that after 30 years of study by hundreds of researchers using the best instruments available, nobody has closed the case in either direction. It sits in a NASA clean room waiting for the next generation of tools. The single most studied object in the search for life beyond this planet, and still no verdict. Number 1. Viking-labeled release. In 1976, the Viking landers scooped up Martian soil and ran the first life detection experiments ever performed on another world. One of those experiments produced a result that researchers are still arguing about half a century later. The labeled release experiment was designed by Gilbert Levin. The idea was simple and sharp. Mix a small sample of Martian soil with a drop of nutrient broth laced with radioactive carbon-14. If living microbes are present, they should eat the nutrients and exhale radioactive carbon dioxide. A detector above the sample would catch that gas and confirm metabolism. Both Viking landers ran the test at sites thousands of kilometers apart. Both gave positive results. The radiation counter climbed in a pattern that matched what happens when you feed terrestrial soil the same broth. When the team heated a control sample to 160 degrees to kill any organisms, the response disappeared. That is exactly what sterilization should do if the signal came from life. But there was a problem. Another instrument on Viking, the gas chromatograph mass spectrometer, failed to detect organic molecules in the soil. At the time, the community reasoned that if there were no organics, there could be no life. Most concluded the labeled release results came from a strange chemical reaction, not biology. Then decades of new discoveries complicated that conclusion. The Phoenix lander found perchlorate salts in Martian soil in 2008. Perchlorates are powerful oxidizers. Lab experiments later showed that heating perchlorate-laced soil in a gas chromatograph destroys organic molecules before the instrument can detect them. The Viking mass spectrometer may have burned the very evidence it was looking for. Curiosity confirmed this. It found organic molecules in Gale Crater using a different detection method that accounted for perchlorate interference. The organics were there all along. Viking just could not see them. Gilbert Levin spent the rest of his career arguing that his experiment did detect life and that the scientific community dismissed it prematurely. He published analyses showing that the labeled release curves matched biological activity and did not match any known purely chemical reaction involving perchlorates or superoxides. The debate is not settled. The positive signal is real. The sterilization control behaved as biology would predict. The chemical explanations have gaps. But without a definitive follow-up experiment on the Martian surface, no one can close the case. No mission since Viking has carried a dedicated metabolism test back to Mars. That makes the labeled release the longest-running open question in planetary science. A 50-year-old experiment whose data still fits life better than any non-biological model put forward to replace it.